All right, welcome back to Bio 1. Today we're going to talk about macromolecules. So we're going to start hooking atoms together to make larger molecules. We call these macromolecules in chemistry and in Bio 1. So what we're going to do today, we're going to talk about four groups of macromolecules. The four basic groups we'll talk about in this class. Many of these you're probably familiar with. We'll talk about carbohydrates. We'll talk about lipids, proteins, and we'll talk about nucleic acids. And what we want to do is be able to learn how to sort of identify them, know what they're used for, and that sort of thing. So let's just start first by talking about carbohydrates. And the thing with carbohydrates is what we'll find is that they generally come in a ratio of 1 carbon for every 2 hydrogens for every 1 oxygen. So a 1 to 2 to 1 ratio. It's not exactly always that exact ratio, but it's always going to be very, very close to that, and you won't confuse that with anything else. So I'll show you what I'm talking about later on. Lipids are mostly carbon and hydrogen, and the most common lipid you probably know about, which is a type of lipid, and we'll talk about that, but those are fats. So we'll talk about that as well. Proteins, you know what proteins are. We'll go into more detail on those today as well as in upcoming weeks. And proteins are made up of smaller subunits called amino acids. So we'll talk about amino acids and what makes an amino acid an amino acid. And then our last group, we really won't talk too much about these right now. We'll save those for upcoming week 8 and 9. But nucleic acids. Nucleic acids are made up of things called nucleotides. And nucleic acids will be things like DNA. We'll talk about deoxyribose nucleic acid, and we'll also talk about RNA. But again, we'll hit most of those later on in upcoming units. So the first one we'll talk about are the carbohydrates, and carbohydrates come in 3 sort of different sizes that we talk about in this class. And the first size are called monosaccharides. So monosaccharides are things such as glucose, which is going to be a very common sugar. Carbohydrates are often, especially the monosaccharides, are referred to as simple sugars. And we'll talk about glucose. So, if you have a friend or family member, or you yourself or a patient or anything like that that has diabetes, one of the things they always are concerned about is your blood sugar level, and that's usually a measurement. We're measuring glucose. In that case, glucose is used as an energy source throughout the body, but particularly very, very important for your brain. And so when your glucose levels go too low, your brain starts having issues, your nervous system, and that becomes very serious very quickly. So when we talk about monosaccharides, what we're going to find is that they typically have 6 carbons, like you see here. So carbons 1, 2, 3, 4, 5, 6. And then you'll notice that in this particular case, it should have 6 oxygens, so that 1 to 2 to 1 ratio, again, that I mentioned, 1 to 2 to 1. So 6 carbons. There's 1 oxygen, there's 2, there's 3, there's 4, there's 5, and I couldn't find the other one, but there it is right there. There's the 6th one. And so there's my 6 oxygens, and then I should have 12 hydrogens. So I've got one there, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now, glucose, you notice this here shows you that this reaction can sort of move back and forth between these 2 states. This is glucose, but this is also glucose. So glucose will tend to form these rings. It'll fold upon itself and form a ring structure like you see here. So both of these are glucose molecules, and you'll notice that in this particular case, what happens is that this number 5 here, this 5 carbon, this is going to fold over and connect here. So this 6 carbon here sticks out, as you see there. And remember that, just to review, that's a covalent bond right there. That's where carbon is sharing an electron with a hydrogen there, and carbon tends to form 4 bonds, so there's 1 there, 1 there, 1 there, 1 there. Okay, so that's glucose. This picture here, I don't find to be a particularly accurate picture. They're trying to make it a little bit too simplistic here, but they're not showing you the other things that would be attached here. But normally, if you see this, what I always tell my students is anytime you see a ring like this, if they don't tell you otherwise, there's a carbon at that corner. So there should be a carbon there and a carbon there. And that's okay to draw the ring that way. But if there's nothing else attached there, you assume there's hydrogens attached there. And you notice that's not always the case over here. And that's why I don't particularly like that picture there. But the ring structures are okay for the most part. The next one then are disaccharides. So we have monosaccharides and then we have disaccharides. And you should know now about this chemical reaction here called dehydration synthesis. Dehydration synthesis. Oops, I messed up there. Dehydration, that should be a T. Dehydration synthesis. And then its counterpart is the reaction called hydrolysis, going the other direction. So dehydration synthesis, you remove the water between two molecules. So here's dehydration synthesis. We remove the molecules and we hook these together. So in this case, we hook glucose and glucose together, two monosaccharides, and we make a disaccharide. This one's maltose, but the one that you should really focus on is when we take glucose, which is a monosaccharide, and fructose, which is a monosaccharide. When we hook those together, you get sucrose. So sucrose is a disaccharide, and sucrose is essentially the type of sugar that's in candy bars and cookies and all that. So this is your typical sugar that you get when you eat sugar. When you think about eating sugar, it's sucrose. Okay, so that's a disaccharide. Hydrolysis, just since we're here, if you go the other direction, like if you have this molecule here and you want to break it down, what can be done is you can add water between these two, as you see here going the other direction, and you can split this. So this could be split from being maltose, could be split into two glucose molecules, and that'll be important as something we do later on. Then our next group of molecules are in the carbohydrate group is the polysaccharides. Polysaccharides are really, really big carbohydrates hooked together with a whole bunch of what we call monomers. A monomer is a single unit, and if you hook a bunch of monomers together, you get a polymer. So a polymer is multiple monomers hooked together. So in this case, glucose is a monomer. And if you hook a whole bunch of glucose molecules together, you get a polymer. And in this case, the polymer is a particular polysaccharide, in this case called starch. So starch, where would you get starch? If you think about starch, you get lots of starch from pasta, from potatoes, and things like that. There are other polysaccharides like glycogen. Glycogen is an energy-rich type of polysaccharide used for storing energy in muscle tissue. So we store up some excess sugar, large carbohydrates, polysaccharides, in this form of glycogen that you can break down into sugars to use later on in your muscles. And then another type is cellulose. And I bring up cellulose now because when we talk about plants, one of the things about plants is that in their cell wall, which they have a lot of cellulose. And cellulose is a polysaccharide that humans have a very difficult time breaking down and digesting, which is one of the reasons why when people go on diets or they just try to eat healthier and get less calories, they eat a lot of plants, because plants, as long as you eat green, leafy, cellulose-rich kinds of plants, it's very difficult to get a lot of calories out of those. Now, if you could eat salad and you pour a bunch of oil and salad dressing on it, that becomes a different story. Okay, our next group are the lipids. And lipids are a very diverse group, many different types of lipids. We'll talk about several of them here. But again, lipids have mostly carbon and hydrogen in them. Okay, and so this is one type of lipid here called fats. That's the one you're kind of most familiar with. And a fat is really made out of two different parts. It's made out of a glycerol that you see here, which is this 3-carbon molecule, and then these long chains that are called fatty acids that you see there. So what happens is typically you'll hook 3 fatty acids to this glycerol, once again using that same kind of reaction, dehydration synthesis, and you'll get a molecule that looks like that. So that's our fat molecule there. And you'll notice that since we're here, if there's a double bond like you see there where 2 carbons are sharing more than 2 electrons, you have double bonds or triple bonds. What happens is it tends to bend the molecule a little bit. So these are what we call saturated fats up here, and then this is an unsaturated fat. So typically this whole molecule would be what you call a monounsaturated fat because it has that one double bond there, and it tends to bend the molecule, and it gives it a slightly different chemical structure and slightly different behavior. Okay, so those are our fats there, and we can talk about saturated fats and unsaturated fats just as I was mentioning there. This shows you just a little bit more there. Something like butter, for example, tends to be a saturated fat, no double bonds, and they tend to be pretty straight like that. But then when you have a unsaturated fat, you have a double bond, you get a bend in there like that. And for the most part, you don't need to memorize this chart down here. It just shows you the percentage of saturated fats versus mono, poly, unsaturated fats in different kinds of things. But the traditional, well, many years ago, it was sort of thought that fats are bad for you. So, fat is bad. You should reduce your amount of fat intake and so forth. And now we've learned more and more that really the bigger problem is saturated fats. Those aren't terribly good for you. But the unsaturated fats tend to actually be very, very good for you. So it's not all fats are the same, it turns out. And so the mono unsaturated fats and the polyunsaturated fats, like in olive oil and things like that, tend to be better for you by far than the saturated fats. And we'll get into a little bit why later on in terms of the cardiovascular system. And recently there's some evidence maybe that some saturated fats are actually good for you as well, at least in certain amounts. Okay. Then we can also talk about lipids a little bit more in the context of molecules such as steroids. Okay, so steroids are another group of lipids. The steroids have these rings that you'll see here. So again, any corner on the ring like that is a carbon with its associated hydrogens. So you notice this is mostly carbon and hydrogen, but all the steroids have these ring structures like that, these 4 rings as you see here. And there are several different types. There's cholesterol is a steroid, testosterone is a steroid, estrogen is a steroid. So these are all different types of steroids. You should be able to recognize, you don't need to know cholesterol from, say, testosterone, but if I give you a structure on a quiz or an exam like this, you should at least know that that's a steroid. And then associated with that, you'll often see like, well, those are your LDL or your HDL, or they'll refer to it as your bad cholesterol level and your good cholesterol level. And I just want to point out that cholesterol, this molecule right here, is cholesterol no matter what. But cholesterol does not travel through your bloodstream very well. It's carried, and it's carried around by certain kinds of proteins. And so what happens is, depending on how the cholesterol is transported through your body, certain kinds of food and what you eat and whatnot will tend to raise what you call your LDL level, your bad cholesterol, which is a low density lipid, low density. Okay, and the low density lipid essentially means that there is the way your body is transporting cholesterol, it's transporting it with a low density type of protein or a group of proteins, and that tends to, we believe, sort of clog up your arteries over time, and it leads to arteriosclerosis, and you can build up of plaques. On the other hand, your HDL level, or your high density lipid, refers to you transporting lipids with a relatively dense type of protein, dense proteins, and that tends to be good for removing plaques and preventing plaque formation and so forth. So once again, it's not your cholesterol itself that we consider just as bad. Your cholesterol, which is one kind of molecule, can be transported in your body in different ways, and it's this high density lipid type of cholesterol reading that you want to be particularly high because it tends to be more helpful and better for your body than transporting it with low density lipids like that. Another group of lipids that are going to be important later on are these phospholipids, and these are a major component of cell membranes. So not cell walls, but cell membranes. Cell membranes are also called the plasma membrane, and that's what you can see kind of down here in this picture, the plasma membrane. And what they do, you notice they have two sort of parts to them. They have this region up here called the phosphate group, which we'll call the polar head. It's polar because things like water can stick to it. Down here, these are the non-polar region. So this is the fat really portion of it. And water, this is non-polar, which means it has no sort of really negative or positive charges, so water does not stick to this part. And so what will happen is when we talk about cell membranes, we'll notice they sort of arrange themselves in this double layer like this. And this is really the fat region of it here, these two chained fatty acids there. And once again, you can see there's that double bond there, and so there's that bend in that case there. But we'll talk more about those later on. The next group are proteins, and proteins are made up of small subunits called amino acids. So you probably heard of amino acids before, and amino acids are called amino acids because they have first this thing called a carboxylic group, which is often associated with something being acidic, and the carboxylic group has a carbon with a double bonded oxygen, and then it also has this oxygen with a hydrogen like that. And so that's my double bonded carbon and oxygen. That's my hydroxyl group there. Collectively, this whole thing's called a carboxyl group. In addition to that, we have the amino group, which is the nitrogen and two hydrogens, as you see there. So that's what makes an amino acid. The side group here means there's something else attached to that. They often refer to that as the R group, so you'll see that over here. But what happens is you notice this part of the amino acid and this part, whoops, go back, there we go. This part of the amino acid right here, and this part here, you notice they're the same on both these molecules. So what makes it different, say serine from leucine, is this side group. So that differs, but this part stays the same. So what happens in proteins is they will be hooked together once again. You can see your dehydration synthesis where you remove the water from it. So here I'm hooking the OH hydroxyl portion of my carboxylic group to the hydrogen there. I hook those together and I form this covalent bond here. This covalent bond is specifically called a peptide bond. So I've hooked one amino acid to another. What happens then is when you hook those amino acids together, as you see in this chain, that becomes what you call your polypeptide chain. So we're going to hook that together, and then what's going to happen is this thing's going to fold up in a three-dimensional shape, and we'll have what we call our primary structure, which is just essentially the order the amino acids are in. And then because certain amino acids are negative or slightly positively charged, they'll be attracted to each other and they'll form hydrogen bonds and things like that. And so what will happen is in the secondary structure, you'll get these folds. you'll get these folds, and there's different kinds of folds in the secondary structure, but you'll get these particular folds because of the negative and positive regions on certain amino acids. In the tertiary structure, more folds get together and you get a longer sort of grouping of those. And then in the quaternary structure, what happens is you get often one polypeptide chain with another polypeptide chain, each with a primary, secondary, and tertiary structure. So when you're done, you get this big blob looking thing of a protein here. And what's important is, we'll get to this later on, but this thing will fold up and you'll get this big blob like structure, and it might look sort of just like a big crazy blob, but what you'll find is there'll be certain parts, like right in here, let's say, where a molecule is designed to fit in, and it will, if it fits in there, it can, say, break that down or split that into two. So we'll find that even though this sort of looks random in like a big giant blob, there's going to be a particular pattern that's very important there for, say, an enzyme. So digesting or breaking down something, this particular protein might have a particular shape that is important based on that structure. So what we'll learn later on is if you change this structure, very bad things can happen. That's where we go with genetics when we get into about the 8th and 9th lecture. Proteins come in different types that you're probably familiar with. We'll start over here with structural proteins. You take things like your hair, okay, and your fingernails. Those are primarily made out of a structural protein called keratin. And so keratin is a protein that's used for protection of your underlying nail, for example. Also, it's an important protein in your hair. A horse, for example, the hoof of a horse that it's actually touching the ground with is primarily the keratin hoof, if you will. So there's some structural proteins. More importantly, though, I think, is when we talk about the biochemistry of life and how life operates and works, we're going to talk about enzymes. So enzymes are going to be important for metabolic reactions. We'll go into much more detail on those coming up soon. And enzymes control the rate at which reactions happen. So we'll talk much more about that later on. And then our last group that we'll talk about are the nucleic acids. And once again, they are made up of nucleotides. And nucleotides come in a couple different types. We're not going to go into detail on these now because we will do so when we get to about the 7th or 8th or 9th lecture. But we'll talk about three important parts. We'll talk about the nitrogenous base. So all nucleotides have this nitrogenous base, they have a sugar, and they have a phosphate group. So those three parts make up a nucleotide, and once again, we'll go into those later on. One of the types of nucleic acids is DNA, and DNA is the genetic program or structure that a cell uses, as we'll find out later on, to make protein. So you, for example, probably know this already, but you look the way you do, and you're a certain height, for example, your eye color, for example, is largely dependent upon, not only dependent upon, but largely dependent on your genetic structure. The environment can influence that, but your genetic structure is the instructions on how you can build proteins to, say, change your eye color, for example. And we'll talk about that later on. And I think that is a good start for macromolecules for now.